When I say tubes and filaments in regard to the cytoskeleton, the only real difference is whether or not they're hollow, okay? If it's hollow, it's a tube. If it's solid, it's a filament. Your book will call them tubules, microtubules, microfilaments. Let's not get hung up on jargon. It's just a network that's almost like a spider web. And I don't mean like the orb weaver spiders. I mean just like that nasty gob of gunk that's up in your corner of your roof where you don't get up there with the broom and sweep it out often enough. We've all seen that, haven't we?
You said, I'm sorry, tube hollow,
Yeah.
filament not.
Tube hollow, filament not.
Okay.
The Conan the Barbarian definition. And I mean that good-naturedly.
Yeah.
Y'all have seen like a cotton ball. It gives shape, it gives structure. If you stretched a membrane across that cotton ball, you've got a kind of pseudo model of what a cell would be. Kind of get an idea how this works. So you can say the cytoskeleton gives it shape, you can say that it gives it support, you can say that it aids in movement. And I see nobody writing any of this down. Uh-oh. You could say that it's an anchor point for various things, because it fills all those roles.
Support structure.
Support structure.
Okay, that's.
And when I say anchoring things to it, you get a chance in lab to see some of the little cells that move. They've got a flagellum, looks like a whip. That flagellum anchors to the cytoskeleton. You see how the amoeba flows into its own pseudopods? That's due to the cytoskeleton. The amoeba is not just a shapeless blob. It's got a cytoskeleton. It can contract and extend and move. You see how it serves as an anchor point? You see how it helps in movement, right? There's not really a lot more to be said about that. If you put support structure on the exam, you'll get full credit, because that's what it is. That's what it does. More than anything. I don't want you putting protection, though, because it's not strong enough to protect in the sense that we think of our skeleton, right? If I fall and hit my head, I've got a pretty good hard bone in there to stop the blow, don't I? If I have somebody swing something at me, I can block it with my arm. It might break the bone, but it's going to still protect me. Cytoskeleton doesn't really do that. It's very, very flexible. Everybody got that?
So more like cartilage.
Huh?
So more like cartilage.
Yeah, something like cartilage. That's a good example. Rubber bands even. Just, it's got some stretchy flexibility to it. It's got some rigidity, but not too much. Good job. It attaches to a lot of different things in the cell. And that's about all we'll say about the cytoskeleton. Next up, I want you to get the cytoplasm. Cytoplasm. The cytoplasm is a viscous liquid. That means it's thick, it's goopy. The viscosity varies considerably. It can be anywhere from, say, warm maple syrup or motor oil, all the way to something just this side of Jell-O. And it plays a dual role, maybe three. Number one, it provides some support and structure in a similar manner to, say, a water balloon. Anybody in here ever had a water balloon, filled it up? It stretches. You've got some shape now to that balloon, don't you? Cytoplasm does the same thing. It basically fills the space and gives it a shape. The second thing it does, and this is one of the more important roles, it allows transport within the cell. It allows for transport within the cell. And when we get into our discussion of cellular transport here in a minute, that'll make a little bit more sense. But make sure you put the word within, or use the term intracellular transport, within the cell. It's not inter, we're not going between cells, we're staying within the cell. Everybody cool with that? So the third thing it does is it provides a location or site where chemical reactions can occur.
It provides, sorry?
It provides a location for chemical reactions to occur. Once you get that, you've got most of the things in the cell. What I'm going to mention now are two very, very specialized organelles. We've already been introduced to one of them, and that is the mitochondrion. The mitochondrion. Mitochondria, plural. As sophisticated as the mitochondria are, they really only have a single function. And this is the only answer that I will accept on the exam. That is, they produce ATP. They produce ATP. Don't tell me they're the powerhouse of the cell. You don't have a little micro generator cranking up electricity in there. It's not a miniature solar panel. But it makes sense, doesn't it? If I told you that everything has to be paid for in cash, no more cards, no more Apple Pay or waving your phone at the magic sensor or whatever y'all do, you'd have to go to the ATM or you'd have to go to the bank, wouldn't you? That is basically the role that the mitochondrion plays. It serves to dispense that energy that the cell can use. Is everyone clear on that? I'm going to talk about the other one, and then I'll talk about their origin. The next group, or the next organelle I want you to know, is called a plastid. A plastid. Plastid. I'm fairly sure that you have encountered a discussion of these before in your high school biology class as chloroplasts. Chloroplasts. Chloro from chlorophyll. Chlorophyll. Chlorophyll is a green pigment. A green pigment. It is found in plants. It absorbs sunlight. So when you look at a leaf, for example, you're looking at something that's nothing more than a giant solar collector. It's absorbing solar energy that the plant will use to make ATP and sugar. So if you wanted to put down the function of a chloroplast in one word, it's photosynthesis. Photosynthesis. Don't worry, we got a whole chapter coming on photosynthesis for me to torment you more, so you'll learn more about that process than you ever wanted to know possible. If it seemed like I'm being a little flip and kind of snarky, I am. But understand this, just about all of the energy that is used by living things comes from this process. There are a few weird things that live deep in the dark places that don't photosynthesize. Travis is like, why are you looking at me?
You talking about my Indian ghost pipes?
I'm getting to those.
Okay.
No, I'm talking about, y'all ever heard of those deep volcanic vents located like 2,000, 3,000 feet down in the ocean? It's so dark down there that sunlight never penetrates. There are critters that can convert that thermal energy into chemical energy that other things can eat and use. But I think we all agree that most of life is found in the lit areas, right? I couldn't even give you a percentage. It's far less than a percent, something like 0.001% of all energy. So photosynthesis, by far the most important role. It's not the only one, though. It's not the only one. There are certain plants that are non-photosynthetic. There's your Indian pipes.
Mm-hmm.
If you want to Google just Indian pipes and see what they look like, they're pretty darn weird things. If you saw them, you'd probably think they were some kind of mushroom. But they are actually a plant. They don't have chloroplasts because they don't have chlorophyll. They lack chlorophyll. Is everyone clear on that? In other words, they're non-photosynthetic plants. And don't worry, I will not go down this path because just because I did my PhD dissertation on these things, I'm sure the rest of you are not that interested. Am I right? Mr. Holt's just kind of going, yeah, I don't think so. Isaiah.
Aren't there some plants that also use a different pigment?
Because if they use a different pigment, it does one of two things. Number one, it allows it to absorb a wider wavelength range of wavelengths of light. Or it might be a reflective pigment to protect something from the dangers of too much sunlight. For example, I've mentioned ultraviolet light a couple of times. Bet y'all didn't realize this, but DNA absorbs ultraviolet light better than any other wavelength. That's one of the reasons why it's so dangerous. If we didn't have the ozone layer, we don't have life on the surface of the earth because the UV light from the sun would sterilize it. That's why anybody tells you that life exists on Mars. No, it doesn't. It might exist under a rock on Mars or in a cave on Mars, but it better be shielded from the direct sunlight. Good question. These things actually have a specialized root called a haustorium that acts like a drill, and they drill directly into their host, and they get everything they need from the host. Pretty cool, isn't it? Y'all familiar with mistletoe? Mistletoe is a parasite. Nice and romantic, huh?
We had some on our tree this winter.
I hope your tree was not living. Was it?
It's a living tree, but a storm actually knocked the mistletoe off.
Okay.
We hope that was all that was on it.
It won't be. If you ever go out, you might even be able to see it just because it's still winter. You know where Stoner and Cooper are down here, right? There's a couple of trees right on the sidewalk there. If you walk down there and look up, you'll see this globular mass of vegetation in there. That's mistletoe. Now, mistletoe is still green. It's somewhat photosynthetic. But the ones I'm talking about, if you Google the Indian pipe or the ghost orchid, pure white. Nothing in there, and it gets everything it needs from the host. Like I say, I think it's pretty cool, but I'll be understanding if you all don't share my enthusiasm. But I want you to understand, those still have plastids. You might be more familiar with something called a chromoplast. A chromoplast. Chromo
It's a
actually.
different kind of, I'm sorry, is this a different kind of organelle or is this just a different kind of plastic?
Different kind of plastic.
Okay.
Don't ever apologize for asking questions.
Well, I don't mean to interrupt you.
Hey, that's what we're here for, the top, interact. Chromo means color, comes from the word chromos, which is Greek for color. Y'all didn't realize that, did you? You thought chrome was the shiny stuff. Anybody here like roses, tomatoes, apples, flowers in general? Chromoplasts, they're storage for pigments, They're actually part of what makes tomatoes, that sort of thing, a very healthy food. And finally, does anybody here like potatoes, potato chips, french fries? All those goodies. They taste good, don't they? Rich in calories. A lot of starch in that bad boy. Well, there is something called an amyloplast. amyloplast, an amyloplast. Amyloplasts store starch. So even though we talk primarily about chloroplasts, because understandably they're by far and away the most important, right? They're not the only form of plastid out there. Is everybody clear on that? Now, one thing I want to remark here, there are several of these organelles that we've discussed that have membranes. The nucleus, the ER, the Golgi, plastids, mitochondria, those 5. They have their own membranes, which means you will not find them in prokaryotes. You will only find them in eukaryotes. Question, somebody was raising a hand. Yes, sir.
Can you repeat that, please?
I can, but I won't. Nucleus, ER, Golgi, mitochondria, and plastids. Yeah, anytime you need something, Nathan, I'll be happy to. Everybody got those? You will not find those in prokaryotes. Travis?
Okay, so on the review, it says no 4 types of organelles. So that's,
Hang on, we'll talk more about that.
okay.
So everybody got that?
So those 5 have membranes.
They have membranes. You will not find them in prokaryotes. I bring this up because I want you to understand that even though eukaryotic cells use chloroplasts for photosynthesis, it does not mean that eukaryotic cells are the only ones capable of photosynthesis. So in other words, there are photosynthetic prokaryotes. There are photosynthetic prokaryotes. The technical term for those is cyanobacteria. Cyanobacteria. C-Y-A-N-O. Cyano. They used to call them blue-green algae back in the day.
Cyto-1.
Bacteria. And this brings us to the probable origin of both plastids and mitochondria. What I'm going to tell you is something called the endosymbiotic theory. The endosymbiotic theory. Endo means inside. Symbiosis means living close together, living in close association. So the hypothesis is that sometime way back in history, a eukaryotic cell engulfed or essentially ate one of these bacteria. And rather than digest and break down the bacterium, it realized, if I supply this little guy with what he needs, he's going to make me a whole lot of ATP. Everybody see how this goes?
Again, sorry,
Yeah,
I didn't get
What did you not
I
get?
didn't get the last.
yeah,
ATP.
a eukaryotic cell ate one
In-
of these.
Ate one of the prokaryotes. The prokaryote made ATP more efficiently. And that's all there is to it. Are you going to keep it around or are you going to kick it out? I'm going to keep it around because it benefits me, doesn't it? I see some motion in the back. I don't have my glasses, so I can't see faces. Are you confused? If so, speak up, because otherwise I will not know.
You're just talking a little fast. My brain doesn't work that fast.
Is this... Hey, never, ever, ever apologize for saying you need me to repeat something or clarify something or go slower.
Yeah, you need you to go a little slower.
We got a eukaryotic cell.
Okay.
Everybody with me? It engulfs or eats a bacterial cell. Everybody with me? Still with me? Now, normally, it would break that down, wouldn't it? Because it's trying to get energy. That's the whole point. But for whatever reason, it not only did not break that down, it decided to keep it around. Because the bacterium that's now living inside the eukaryote makes ATP more efficiently than the eukaryote could on its own. Does that make sense? Now, anybody here own a dog or have owned a dog, love a dog, raise your hand. A lot of dog people could. Why did humans domesticate dogs?
Hunting.
Primarily hunting. We have to feed that dog, don't we? Does that cost us money? But I'd be willing to bet none of us would willingly give up our dog because we get a lot more benefit from the intangible, don't we? Especially if you're a dog person, you love your dog. But when dogs were first domesticated from wolves, it actually meant both parties were benefiting. The humans had to share part of what they got from the hunt, didn't they? But the benefit to the dog of having a regular feeding schedule was so beneficial that the dogs kept hanging around the camp. They didn't mind doing a little bit extra because they were guaranteed food. And that continues to this day. So does everybody see how symbiosis works? Yes, sir.
I heard cats kind of domesticated themselves.
You heard cat what?
I heard that cats kind of domesticated themselves because.
Have you ever owned a cat?
Yeah, I own
No. The cat owns you.
a cat. True.
Cats are a bit different insofar as cats basically hung around humans, not because humans were sharing the kill so much as humans were storing large amounts of grain in pits in the earth or in small buildings. That's basically a food bank.
Yeah,
It's going to attract things that eat grain, right?
yeah.
Rats, mice. What do cats eat? Stalk and enjoy killing. Rats, mice, yeah. Birds, any kind of small game. My neighbor's cat kept presenting me with its latest kill. Okay, thank you, Sammy. I don't want that dead bird, but I sure appreciate you bringing it to my front porch.
We had an outside cat that decapitated a chipmunk and
Not
brought the
surprised.
decapitated body to the door.
I'm not surprised.
Mhm.
But even that right there is a degree of symbiosis. We're not talking domestication, we're talking living in close association. Did humans benefit from the cats being around?
Yeah.
Yeah, because
Yeah.
those rodents are no longer eating our stored grain, are they?
Yeah.
The cats benefit from the humans and their activity because it's attracting the rodents. Everybody get an idea what symbiosis works like?
Yeah.
So now, we've got this captive bacterium living inside our cell that makes ATP for us. Does everybody see how it's endosymbiosis? Then, sometime a few million years later, one of those eukaryotic cells manages to capture another bacterium, but this one is photosynthetic. It makes its own food. I don't know about you, but I'm gonna put that bad boy to work making me some food too. Do you think so? We've already got one making ATP, let's give him a buddy. Now he's making sugar along with ATP. Right? Coincide.
Okay, so you
That,
carried,
I'm sorry, what?
you carried, captured another bacterium.
But this one was photosynthetic. Well, the first bacterium becomes mitochondria. All eukaryotic cells have mitochondria. That's why we think it came first. We're not photosynthetic, are we? So the line that led to plants captured their pseudochloroplast a little bit later. Everybody see how that goes? And that is the endosymbiotic theory. And before you think I'm up here spouting a lot of caca, there are actually multiple lines of evidence to support this. Number one, both mitochondria and chloroplasts contain their own DNA. They contain their own DNA.
What contained their own DNA?
Huh?
What contained their own DNA?
Mitochondria and chloroplasts, or plasties. Even those non-photosynthetic guys we told you about, they've got plastids. Those plastids have their own DNA. And number two, that DNA is actually more similar to bacterial DNA than to eukaryotic DNA. It's more similar to bacterial DNA than eukaryotic DNA. Everybody still with me? Okay, what do we got? About a page of new notes so far?
Mhm.
Yeah.
Okay. Next up, we've got specialized organelles that break things down. They break things down. These are called lysosomes, Lysosomes, lysosomes. And peroxisomes, peroxisomes, peroxisomes. Basically, these are just water balloons inside the cell. They have their own membrane, but rather than serving a specific function, they're more or less a way we can store dangerous things. Lysosomes store digestive enzymes. Peroxisomes store toxins and then break them down.
Lysosomes store.
Toxins, poisons.
So they both
It-
store toxins.
No, peroxisomes store toxins. Lysosomes store digestive enzymes.
Poison.
Poison is fine, absolutely. I know there are technical definitions that differentiate between toxin and poison and venom, and I don't care. This is not.
I'm gonna say, I'm looking at the definition of it right now. Strong molecules are antioxidants that form in the cell. This is like, it detoxifies.
Yeah. So if you ingest something or the cell engulfs something that's toxic, it'll put it into a peroxisome and break it down. If the cell ingests something like an amoeba engulfs food, it doesn't have a digestive system, does it? Because it's a single cell. What'll happen is a lysosome will attach to that, it'll release the digestive enzymes and break it down. It can even digest itself. If you've got a cell that commits suicide, we talked about apoptosis, you'll want to recycle as much of the material in that as you can. It will actually open up its own lysosomes and they'll start digesting the cell itself. And that's it for organelles. Does that sound good to everybody? So next up, we've got three structures that are related. The first is the cell membrane. Essentially, it controls what goes in and out of the cell. Do I want to control who comes in to my classroom? I may want to control who goes out of my classroom, right? Looking at you, Nick. Joking aside, it makes it easier for us to learn, doesn't it? Because we're not being interrupted by people wandering in here who don't belong. If there were some danger, there's a measure of security. I could lock the door, we could all get back in the corner there and be out of the way of sight and sound, right? The cell membrane is the same way. It's a barrier that controls access. It is a barrier that controls access. Pam was a little late today, but I let her in because she belongs in here, right? And I'm not picking on you, Pam.
It's fine.
What was the tagline on this? Three something that's
Three
real.
related structures.
Three related structures.
Thank you.
And the thing about the cell membrane, I want you to understand, it's not an impenetrable barrier. It's a selective barrier. Pam is enrolled in the class. She belongs here. I'm going to let her in, right? My crazy ex-wife shows up. Do you think I'm going to let her in? That would be a no.
You have a crazy ex-wife?
Huh?
You have a crazy ex-wife?
I do.
What happens if somebody else lets her in?
What happens if somebody else lets her in? Life gets interesting. And we discovered the meaning of that old curse. May you live in interesting times. Because we will be in interesting times. And then we'll see why it's a curse. We don't want somebody who's not belonging in here, right? Does everybody see that? That's the function of the cell membrane. And that's all you have to get down is that it controls access, in or out. Now, you know, I don't mind if you get up and walk out, run down to the bathroom, or you get yourself a drink, or you answer a phone call or something. But if I had the whole class walking up, I might go up and kind of block the door and say, hey, what's going on, right? Again, I'm controlling access. Not saying I wouldn't let you go out, but I'd want to know why. One person not worried about it. I lose half my students or more in a mass exodus. Yeah, that's something of concern. Same principle. You're not only controlling what comes in, you're controlling what goes out. Because you don't want to lose resources that you need for function. If you are in an area where water is scarce, you don't want water leaving the cell, do you? You want to hang on to it. If you've got more sugar inside the cell than outside the cell, you want to hold on to what you've got, don't you? You may want to bring some additional sugar in. And that's what the cell membrane does. Next up, you've got the cell question.
No.
Okay.
I'm sorry.
Well, it's not your fault. Like I said, all I see is a blurry something. I just saw motion. I didn't know if somebody's raising their hand or what. Are you doing something back there?
I'm itching my head.
Okay. I don't know if she's really raising her hand or if she's just messing with me.
I just fidget a lot.
Cell wall. Not every organism has a cell wall. The biggest group that doesn't are animals. We don't have a cell wall. Plants, fungi, protists, prokaryotes, they all can have a cell wall. The cell wall, unlike the plasma membrane, isn't flexible. It is rigid to a degree. It is hard. Its real function is to provide support, to a lesser degree to provide protection. Support and to a lesser degree protection. Cell wall is fairly rigid, but generally it retains some flexibility. If you've ever had a green twig, you know you can bend it a little bit, right? It's got some flexibility to it. If you push it too far, though, it breaks, right? It can be hard. Wood, for example, has a rigid and hard cell wall. And finally, don't write this down unless you really want to. We'll just use the abbreviation. We have what we call the ECM. I do want you to get the abbreviation, the ECM. The ECM stands for extracellular matrix. You don't have to write that down. If you use ECM on the test, that's fine. Question? The ECM are just molecules that stick outside of the cell wall or cell membrane or both. They're molecules that originate in the cell but stick out beyond it. And they basically function like glue. They allow cells to stick together.
Does that mean single-celled eukaryotes? Does that mean single-celled eukaryotes don't have it?
Nope, that doesn't mean that,
Oh, it doesn't?
because I'm getting there. Like always, you just want to jump ahead. He'd be a good scout in the military. He's gonna fall in the pit. The other thing it does is it serves as recognition. Your immune system needs to know if something belongs in your bloodstream, doesn't it? If a white blood cell bumps into a red blood cell, you don't want it eating the red blood cell, do you? Because the red blood cell serves a function in your body. It belongs. So when those ECMs of each of those come into contact, it recognizes it as belonging. When it bumps into a bacterial cell, that's foreign, isn't it? It does not belong. So we're going to throw rocks at it until it leaves, right? So if I go out and I get my UT orange jacket and I wear it in here, Isaiah's going to throw rocks at me until I leave. If I wear a blue jacket, we're all going to say, well, this is Kentucky, we're all Cats fans, right?
Nope.
We'll stand against them. Does everybody see the, I know it's kind of joking, but you see how it works? And that's why single-cell eukaryotes, they do that. Because if they come in contact with another one of their species, they don't want to fight it, engulf it, or anything. Everybody got that? Last thing we'll talk about, as far as organelles and all this, transport. Transport across the cell membrane.
And that's what HCM also does?
No, this is just the cell membrane. The ECM, we're done with the ECM. It just does recognition and glue, allows things to stick together.
So we're going back to cell membrane.
We're talking about the process of transportation across the membrane.
Okay.
Let's say I opened up a can of skunk scent and set it on that desk there. Who's gonna smell it first? Huh? Who's gonna smell it first?
Me, you.
Probably Pam, probably me. Who's gonna smell it next? Then you. All you over there in the back row and over here on the left going, yeah, we're glad we're far away from it, right? That is because those scent molecules are moving down a concentration gradient. They are moving down a concentration gradient. They are going from an area of high concentration to an area of low concentration. Does everybody see how that goes? If I took a glass of water and I put a drop of food coloring in it, let's say red, I'm gonna have a very intensely red dot, and the rest of it's still gonna be transparent, isn't it? If I come back in about 20 or 30 minutes, the whole glass will be a pale red, won't it? Because it's equal color. You see how it's gone from high to low? That is called diffusion. Diffusion. You go from high concentration to low concentration. It does not require any energy. It does not require any energy. We call this passive transport. Passive transport. Eventually, it will reach a state of equilibrium, right? Where we have an equal concentration everywhere in our container. Our container being the room. We said the door was like a cell membrane, right? That's a selective barrier, isn't it? Now, if I open that door, the people down the hall in the physics lab aren't gonna be too happy with me, are they? Because sooner or later, they're gonna start smelling that skunk, aren't they? See how it goes? By the time it gets all over to Dr. Castle's office, hopefully we'll be evac. So otherwise, we might really have some issues. You get the idea, though. It expands to fill the container until it reaches equilibrium. There's a related term called osmosis. Osmosis. O-S-M-O-S-I-S. Osmosis is simply the diffusion of water.
Is that why the fancy bottles of water say, process of reverse osmosis?
Yeah.
Or purified through reverse osmosis or something?
Isn't there a movie?
Osmosis Jones.
That movie kills me.
Well, that's a cool sounding name, isn't it? I can't tell you the number of names where I see they mislabel things. So is everybody cool with that? The main thing I want you to understand, you go from high to low, it doesn't cost energy, and it's passive transport. The reverse of that is called active transport, because we're going to have to spend energy to make it happen. Let's say I had a cup full of ball bearings. Everybody knows what ball bearings are, right? Little marbles. If I tip that cup over, they're gonna roll all over the floor, aren't they? And then I make you pick them up. Y'all are gonna have to expend some energy to do that, aren't you? Every one of you is gonna have to bend over, pick it, bring it up here, drop it in the cup, go back, pick up another one, bring it up here. You see how that's going? Are we concentrating the ball bearings? We're going from low concentration to high concentration, aren't we? But we have to expend energy to do it. That's called active transport.
So it requires energy for diffusion?
No.
No.
It requires energy for active transport. Diffusion is passive transport. Active transport is the opposite of diffusion.
Active transport requires energy to go from a low concentration to high concentration.
Yep. Imagine I've got my car sitting on top of the hill. And I leave it in neutral. Slightest push can send it down the hill, right? It's going from a higher energy state to lower. Does everybody agree? You really don't have to do anything, especially if you park on the side of the hill. You just get out and leave the brake off, it's going down. It wants to. That's how diffusion works. High energy state to low energy state. High concentration to low concentration. But I like making my students suffer, and I want the car at the top of the hill. So instead of giving you my keys and letting you start the engine and drive it up there, I'm gonna make you all push it up there. Is that gonna cost you energy?
Yeah.
Even if we drove it, it costs energy, but it would cost gasoline. Even if we drove it, it would cost energy.
For the birds and gas to get out. Does everybody see that? I'm happy to extend energy to get from the lower state or the lower concentration to the higher state or the higher concentration. Now, why would a cell want to do that? Well, let's say that each of us represent a sugar molecule and the four walls of this room represent a cell. We have to break sugar down to get ATP, don't we? Am I gonna eventually run out of sugar? So the next person that comes through that door, I'm gonna step outside and thump and drag them in. Yeah, you get broken down. See somebody wander out one of those rooms, boom, I nailed you. I'm definitely gonna be controlling access. I'm not letting you out of this room yet.
Good.
Everybody see how that works? Because we have a higher concentration in here than probably in the hall right now, don't we? But I still wanna get that one out of the hall and bring it in because I wanna use it eventually. So does everybody see the difference? It's not hard, is it? There is another form called semi-active. Semi-active transport. What happens with this, it wants to go from high to low, but it can't because there's a barrier there. Everybody see the marker in my hands? It wants to fall to the surface of that desk, doesn't it? Can it do that as long as my hands are like this? Because they're forming a barrier, right? I expend energy to open up my hands. Did I expend energy to force the marker down?
No.
It did that on its own, didn't it? But I had to open the gate first. Semi-active is basically diffusion, where you have to actually physically open the gate, which costs you a little bit. Everybody cool? Does that sound like a good place to stop? Okay, now, some things to add to your study guide. Give everybody a chance to get out something to write on, something to write with. We did talk about the cell cycle, didn't we? So I would strongly suggest that you be able to draw or describe or explain the cell cycle. You might want to know the difference between G1 and G0. I think I told you that, didn't I?
Did he?
Yeah.
How long ago? was
G1,
this?
what's that? G1?
No.
Growth 1. Growth 1 or growth 0.
Okay. What did you say?
Related to that, you probably want to know the term differentiation. Differentiation, which means the specialization of a cell. Related to that, I believe we talked about 4 different kinds of stem cells, didn't we? Totipotent, pluripotent, multipotent, and unipotent, or monopotent. Y'all remember that? You remember the relationship between specialization and how well it can divide? The more specialized it is, the less likely it is to divide. The less specialized it is, the more likely it is to divide.
4 different kinds of
Stem cells. Yes, ma'am.
stem cells.
Differentiation was the specialization of a cell.
A sperm cell is very different from a blood cell, isn't it?
I'd say so.
Huh?
I'd say so.
Yeah. That's differentiation, specialization.
You said no, the G1 and G2 or G0?
Zero.
The cell cycle, the whole cell cycle, G1S, G2M. But if it never leaves the G1 phase, we don't call it G1, we call it G0. So a specialized cell is gonna stay in G0 and it will not divide, right? Know the Hayflick limit,
I'm sorry? No.
Hayflick limit. What causes the Hayflick limit? Telomeres get shorter, telomeres get shorter. Know the enzyme that allows them to get longer again, telomerase. What are the two types of cells that express or make telomerase? Nobody can tell me?
I,
I don't remember this.
I'm sure we'll keep up.
Cancer and embryos. Cancer cells and embryos, remember HeLa? It's just last lecture, oh dear.
What was it, cancer cells and what else?
Embryo.
Okay, you said know the Hayflick limit and then what?
The two types of cells that get away from that are embryos and cancer, and they get away from that because they make telomerase, which makes the telomeres longer rather than shorter, which is what normally happens. Know the 4 stages of cancer. Know the 4 stages of cancer. No cancer treatments. What's the preferred treatment of cancer?
Surgery.
Surgery. What are your backups?
Radiation.
Radiation and chemo. And if you don't know these, that's okay. This is, remember, it's review. As long as you write it down, you'll know to look it up. I will also make sure that I check my email extensively over the weekend. And I think several of you have already sent me emails and you found out I actually do answer them, don't I? So if you have a question and we can't explain it via email, what should you do? Tell me you're not understanding it. And I'll say, okay, Ned, you got my phone number, give me a call, and we'll chat 5 minutes and you'll understand it. Ned says, I still ain't getting it. I'll say, okay, do you have Teams on your computer? And I'll say, nobody uses Microsoft anymore. And I'll cuss under my breath, and then I'll download FaceTalk or FaceTime or Time Talk or whatever the heck it is, all you Apple users use. Right? Joking aside, seriously, I do not mind if Sunday afternoon I get an email saying, I'd like to talk to you, can we do a Zoom meeting and chat about this? I can't do as much as I can in person, but I've got a little whiteboard that I actually used when I talked during COVID. I might dig that out and we can hold it up to each other's cameras and just get all 21st century. Everybody cool with that? Any questions about the exam, the material on it, or when we have it?
You said there's 10 questions on the exam.
Generally 10. It might be 8, it might be 10, it might be 12, but I generally try to shoot for 10. My rule of thumb is I basically give myself 10 to 15 minutes to take the exam. Meaning I can write the answer down in a format that is completely acceptable. If I can do that in 10 to 15, it usually means you all can do it in 45 to 60. That leaves us a little bit of extra time left over. Does that sound good to everyone? Now, I don't have anything after this class except a 1-hour drive home. So if you are not done at 3:15 and you buy me a soda, I'll sit here and drink it. No, you don't have to buy me a soda. If you still have a question or two going and it's 3:15, I'll still stick around. That sound good to everyone? What are you gonna do if you wake up at 3 in the morning, Sunday night, and you're freaking out with anxiety? What are you gonna do about this test?
Drink.
What'd you say?
Drink.
You're gonna email me and say you're freaking out and you're not gonna make it to the test.
Well, you better drink when you study because you need to be in the same
Drink
state when
water.
you study than when
Water.
you take the test.
Yeah,
So.
and I certainly hope you're not taking the test drunk. I've seen enough of that at Vanderbilt. I've told you so much about those places already. They have a drinking problem there. Okay, here's what I want you all to do with your remaining time. I want you to come up with 3 potential exam questions, write them down with their answers, and turn them in. Once you get that done, and you put them right here in the corner of my desk, you're free to go. If you have lab with me, you have to stick around until 3:30. You may work together, you may ask me, or use the books, the notes, the review, anything you want to. It's just a little exercise to help you get to thinking about this, okay? I can't see you. You frowning at me? He's not even looking at me.
I do have a question
Yeah.
about like the question earlier. Did you say short answer or?
Yeah.
So we can,
Could it be like one
Trigger
sentence, like?
question 1 before the 2 laws of thermodynamics, sentence proof.
yeah, do that.
We can do
So, what are the